Hello, my name is Sarah, and today I will be discussing my research done over the perceived effectiveness and administration of anthelmintics in beef cattle in relation to the actual parasite load. The primary purpose of this research was to educate producers on the effects of anthelmintics, which are any drug that acts against parasitic infection, compared to the actual parasite load found within their cattle. The type of parasites we will be discussing are internal parasites, any parasite that lives in the gastrointestinal area of a bovine. There are four main types in our region of Missouri. Those are nematodes, hookworms, roundworms, and tapeworms. So why are parasites a problem? Why should we do research in this area? Well, any mature parasite that lives within a host can lay up to 10,000 eggs per day. These eggs are excreted via a dung pad, which does sound a bit disgusting, but it's actually the optimal environment for the eggs to be able to hatch and the larvae to be able to infect a large area of the pasture. Once the pasture has been infected, the grazing bovine will then orally ingest the parasitic larva. And once the larva has been ingested, it takes about 21 days for this parasite to mature, and then it can start laying those 10,000 eggs per day. So parasites are effective at reinfecting a host and infecting many hosts within a given area, especially host-dense areas such as herds. So once a parasite is living within a cow, what are some impacts that we see on the host? Two main areas of impact. The first is the health of the bovine. Early symptoms of a parasitic infection tend to be subclinical. Thus, it is not very easy to see that the cow is suffering from this parasitic infection, because most early symptoms include disruption of absorption and digestion of key nutrients, causing to mainly severe stomach aches, inability to absorb things well, which ultimately causes a slow decline in weight gain. However, after 5 months of the parasite living within a host, what we see is a high increase in both size and number of parasites. And that's where we start seeing the critical damage that causes severe clinical issues, such as severe dehydration, anorexia, enteritis, which is swelling. Reproduction capacity reduces, and in extreme cases, we even see death due to these parasitic infections. Because early symptoms are subclinical, more often than not, a bovine will go undetected as having a parasitic infection until there's already been critical destruction and the weight gain is severely lost. And that's where we start getting into the economic problem, where cows are sold by pound. Thus, producers want to have as heavy and as much weight on a cow as they can. But parasites will cause anywhere from a 10 to 20% loss in weight for each bovine it's infecting, which can be anywhere from $600 to $1,200 in market loss per head of cattle. Overall, it is suggested that about $3 billion are lost annually in the ranching industry due to parasitic worm infection. Thus, it is a problem, and steps have been taken to try to reduce this problem. And that's where anthelmintics come in. These have been around for a while. These are drugs that act against parasitic worm infection. Their main goal is for the producer to be able to gain control over the parasite load. Our research was looking into these anthelmintics. We had 3 areas of focus. Type, which there are 3. We have pour-on, injection, and oral. The only difference between these is the type of administration. We also looked at the frequency of the treatment and then the rotation between the types of treatment. Now, this last point, the rotation, is very critical for our research because of something that's been surfacing over the last 5 years, and that's anti-parasitic resistance. Because parasites have such a high reproductive capacity, what we see is microevolution occurring within these parasitic populations. So that specific traits that cause these mature parasites to survive an antihelmintic dose, they're passing on to their offspring, who are then adapting and passing those things on to their offspring. Therefore, we see an increase in the number of parasitic parasites that survive per population after an antihelmintic dose. We see a decrease in duration and effectiveness of antihelmintics, and in some cases, we see complete loss of effectiveness. So these two things have been utilized. Antihelmintics are utilized in the ranching community, and the scientific community has been looking into anti-parasitic resistance. However, there is no research done to bridge these two to see how anti-parasitic resistance is affecting antihelmintics, specifically how anti-parasitic resistance is being affected by rotating antihelmintic processes. And that's where our research is hoping to bridge a gap. For we believe that the actual parasite loads will be greater than perceived by each producer based on their antihelmintic process due to a knowledge gap in the effects of type, frequency, and rotation of antihelmintics used, mainly based on anti-parasitic resistance occurring. So to move into our methods, essentially what we needed to do to test our hypothesis was to get a fecal egg count in correlation with the antihelmintics used. To do this, we collected a fecal sample. We collected these fecal samples fresh, immediately bagged, tagged, and put them on ice. This reduced their contamination. We also administered a survey to each rancher. This survey included information such as their treatment choice of antihelmintics. Do they think it's effective? Why do they use it? The frequency of their treatment, and whether or not they rotate their treatment. Once we had these things, we headed back to the lab. Once in the lab, we were able to make a density gradient solution that allowed us to actually count the number of eggs within the sample. We did this by measuring 4 grams of fecal matter and mixing it with a flotation solution. Once mixed, we ran the solution through a strainer to remove any large debris, which essentially left us with a solution that was primarily the parasitic eggs from the sample. Once this solution was allowed to sit, because of the density gradient nature, those eggs would float to the top of the solution so that they could be observed under a microscope. This solution was added to a specific type of slide called the McMaster slide. This slide had a grid in it. So once the solution was filled, you were able to go up and down each grid mark and calculate the number of eggs you observed. Using this number, we were able to calculate the eggs per gram of fecal matter, which gave us the parasitic load of the herd in question. We were able to pair that number with the survey questions, which allowed us to perform an ANOVA test based off parasite load and antihelmintic used. An ANOVA test is a multiple mean comparison, so we're able to compare each of our group samples. And once that test was run, we were able to pull some results. Overall, based off our survey questions, we were able to group all of our samples into 4 different types of groups. Number 1 was our no treatment group. These samples had no antihelmintic treatment. And we had our group 2, which is our poron antihelmintic group. Both of these groups act as a control. Group 1 acts as a control to see the impacts of antihelmintics in general, whether or not you rotate or don't rotate. And then group 2, which simply had one type of antihelmintic, poron. This one acted as a control to see the effects of not rotating versus rotating. And then our last 2 groups, group 3 and group 4, were both a rotation group between a poron and an injection, with the only difference being group 3, which is our rotation plus. This had a specific and more expensive type of injection called the long-range injection. We were then able to use these groups to graph some results. And we get this beautiful graph. This graph allows us to see the comparison between the mean A count, which is our parasite load that you can see on that y-axis. As it goes up the y-axis, you're increasing in your parasite load. And then on the x-axis, we see the different types of treatments. Our red is our group 1 or no treatment. Our orange is our poron or single anthelmintic treatment. And then you have your blue, which is your rotation plus, and your purple, which is simply your rotation. Both of those rotations were a poron and an injection rotation. One more thing I'd like to draw your attention to on this graph is the letters above each right-hand corner. Because ANOVAs are multiple mean comparison, we are comparing means in this graph, and those letters represent the means. Each letter is statistically different than the other. So our X mean of our red no treatment group is significantly greater in parasite load than our orange group, who has the Y mean, and significantly greater than our Z group. And then we move down to our Y group. As we mentioned, it's lower than our X group, but it's also almost double our Z group. And then both Z's are used to represent the means of both rotation groups because both of these groups were statistically identical. So now that we know the general trend of parasite loads, what does this actually tell us? Well, here's the significance. Starting with group 1, this is our no treatment group. This was our control. As we could see from that graph, it was significantly higher parasite load than any of the other treatments. These ranchers did not achieve parasitic control. Therefore, the perceived effectiveness of their treatments did not correlate with the parasite load found within their cattle. And it is suggested, based off their parasite load, that these cattle be treated for parasites, or their cattle may suffer parasitic illnesses. What this group showed us was that whether you use a single anthelmintic or rotate anthelmintics, you will have significantly less parasite load than if you do not use any type of treatment at all. Now, the most interesting results came from our group 2. This was our poron only treatment. Interestingly enough, this group also did not achieve parasitic control. Thus, it is suggested by professionals that these cattle get treated for parasites to preserve the health of their bovine. Therefore, poron only did not achieve what the ranchers believed they were achieving with this treatment. Furthermore, poron is the most widely and longest used anthelmintic treatment. Thus, it had a longer duration time for parasites to be able to build up more and more tolerance and immunity to it. Thus, our group 2, based off our experiment, suggests that anti-parasitic resistance is occurring, specifically with no treatment rotation, as well as long duration of the same type of treatment, to the point where parasitic resistance is causing the achievement of parasitic control to fail. These ranchers' treatment did not work. And then we move on to our rotation groups. We clumped them together since they were statistically the same. We saw that they did achieve parasite control. Thus, rotation, as suggested from our research, increases the effectiveness of the anthelmintic. Overall, we saw that treatment is beneficial whether you rotate or not. But if you do not rotate with a long duration of the same type of treatment, you lose control, and rotation is beneficial for increasing anthelmintics. There are 3 areas we hope to improve. We hope to educate producers so they can better benefit in their business. We hope to springboard further research, and we hope to increase bovine health. Thank you to all of the following.